Literally share it in the group again. Bioethics and CAD. Thank you everyone who's joined. Thank you for being on time. We really appreciate it. It allows us to go as per planned. So thank you again. Shubham, if I'm not wrong, Dr. Richa has still not joined, right? You're muted, Shubham.
Texted ma'am. I also asked ma'am to join. She said she'll need more 5 minutes.
Okay, no problem.
Yeah, yeah. Mary Matthew ma'am is also. I think Richa Dayaramani ma'am has joined, if I'm not wrong.
Yes, I'm here.
Okay, ma'am. Thank you so much.
Mine, sorry to, matlab, my name is Dr. Richa Dayaramani.
Dayaramani.
We'll keep that in mind, ma'am.
Okay,
Sorry.
okay, okay.
Sorry,
Sure.
that's okay. I mean, but
Yes,
doesn't
yes,
matter that
definitely.
much. Hello, ma'am.
Ma'am, you're muted. Princy, ma'am, you're muted.
देखो वो देना उसको ऊपर करनी
You're still muted, ma'am.
David,
Good evening, Mary ma'am. We are so glad that you are along with us.
I'm muted. Can you hear me?
We can, ma'am. We can hear you.
Hi, hi. I'm sitting in the car.
Oh,
Where's Dr. Russell hasn't joined?
I'm waiting for Sir to join. I have texted him.
Okay. No, there's 2 minutes more, so probably he'll join in the last moment. What's the problem, Princy? What is it?
no, nothing.
Okay.
I'm fine. Everything's good now. I just wonder, Anusha, can you see my PPT screen?
No, ma'am.
Oh, ma'am. You have to
No,
present.
I haven't shared. You shared it from your end.
Yeah, Jalak, are you here?
Yeah, hello. Yeah, hi.
Could you share this?
Wait just a second.
I'll call Sir once again and see what's.
Fine.
Do you see the screen?
Yeah, visible.
Sir is also joining the call. Sir said he'll be joining in a minute. Is Russell sir with us?
Okay. Yes, I've joined you.
Okay, sir. We'll be starting with that.
Good day, Princy. And where is Dr.
Yes.
Mary?
Good evening, sir.
Okay,
Mary ma'am has also joined the call.
very good.
Keegan and Anusha.
And
Good evening, sir. How are you?
Good evening,
Good
sir.
evening.
I think we should start now.
yes, hello, Anusha. And yes, so who else is there? Okay.
Anusha.
Yeah, good
Should we
day, Richa.
start?
Yes.
Of
Yes.
course.
Go ahead.
Right. A very warm welcome to all of you present here today. We're so grateful to you for coming and joining us this evening. I'm Rotractor Anusha Prabhu.
And I'm
Keegan,
Rotaract Keegan Miranda.
and we're super happy to host you this evening. We also have with us students from the Medical Student Association, MSAI, and
And we also have students from Asian Student Medical Association, that is AMSA.
so a little bit about Rotaract Club of the Caduceus. So, like all of you know, this is Rotaract club. This is a Rotaract club that's specifically for medical students from all courses, that is BDS, MBBS, physiotherapy, etc. And we organize a lot of webinars and outreach programs that are basically medical camps to reach out to those who are less fortunate. And we've been working pretty hard even during the quarantine. And if I speak for myself, and I think I can speak for Keegan as well when I say this, more than anything, Rotaract has given me a purpose. Because as medical students, we are locked in a cocoon with our books, oblivious to the external world. So engrossed in our studies, and we don't get the kind of exposure that you really need as medical students. To make a difference and to go and serve. And our theme for this year is Revolutionize, that is dedicated to discover, created, and committed to care. So we urge all of you to join us in our movement to make a difference to the masses and reach out to as many as you can. We wouldn't want you to miss out on the opportunities that we're getting at this stage in our lives. So we'll be dropping the link to register for Rotaract as well in the chat box. We urge you to give it a look and check us out. Coming to bioethics, as medical students, we've realized how crucial this is in our day-to-day life. We come across situations where you don't really have black and white areas, and in these gray areas, quite often we don't know which way to go. And bioethics basically gives us a better idea of which part to take in really controversial situations. It's there to protect both the patient and the doctor and to ensure that the right care is given to the patient and the doctor does justice to the particular case at hand. So we're really excited to bring to you a 3-day conference that is today, tomorrow, and day after. And we hope you continue to tune in and learn with us through this process.
Great. So a little more about our conference and what we intend on doing in the span of these 3 days. We have already introduced to you our conference and its name, that is Bioethics Colloquium at Caduceus. And we're doing this in association with some of the biggest medical organizations in India as of today. We're doing this with, as Anusha said, MSAI, that's the Medical Student Association of India, and AMSA, which is the Asian Medical Student Association, along with IMA MSN, who will be joining us on our last day for the debate. So just to get some of the doubts away and some of the concerns away, I will give you a small gist of what we're going to be doing on these 3 days. So today we have a panel discussion by a very, very, very experienced panel who we will be introducing later on the front of CRISPR Cas9, the ethical dilemma that comes along with this new revolutionary miracle technology. Okay, so tomorrow we have Tomorrow we have another such panel discussion. We on another topic which you will find in our brochure that we've circulated almost everywhere. After today, we have a competition that is the photo elocution competition. So if you haven't taken part already, today is the last day to send in your entries. So make sure you do. We will be sending you out. We will be sending out the messages again if you want to understand what the competition is about, so that you give your best entry and give it your best shot. And lastly, to as a cherry on top of the cake, we have a debate. The debate is going to be between four huge medical organizations in India. We have MSAI, AMSA, IMA, and Caduceus, who will be battling it out with a legal simulation of a case with special emphasis on the bioethical aspect of it. And we have amazing judges, we have amazing panelists, we're just blessed with so many amazing people to join us. And I'm sure that each one and every one of us is going to learn so much out of these 3 days. So, with that, I will introduce our college representative from DY Patil Kolhapur, uh, Rotaractor Shubham Gupta, who will be introducing our wonderful panelists to you. Thank you. Rotaractor Shubham Gupta, do we have you?
Yes, yes. A very good evening to one and all present here. We are delighted to have all the esteemed panelists from India and international today. They will be sparing a precious time with us today. So let me introduce our esteemed and guest of honor, Dr. Russell D'Souza. Dr. Russell D'Souza, Professor Dr. Russell D'Souza is currently the Executive Director Global Operations TOITM, International Institute of Organizational Psychological Medicine. In 2011, he was appointed the Chair Asia Pacific Bioethics Network for Education, Science and Technology, mandated to establishing UNESCO Bioethics Unit in universities and center in Australia, the Pacific and Asia that will respond to emerging trends in bioethics generated by the UNESCO as expressed in its Universal Declaration on Bioethics and Human Rights, October 2005. The Asia Pacific Network works to strengthen, develop, promote, and carry out academics, research, training, as well as other activities toward achieving the UNESCO objectives. Sir was also appointed to the scientific section of disaster psychiatry of the World Psychiatric Association. He has contributed to psychiatry, mental health services, and rural health services in Australia and worked in Australian public health system for last 25 years. Professor Russell D'Souza was elected the founding president of Indian Global Psychiatry Initiative. Sir, I would like you to please address all the participants today and also tell some words of wisdom. Over to you, sir.
Thank you, Shivam, and thank you to the Rotaract. I had the opportunity of meeting and discussing, coming to learn about Rotaract from Anusha and Kagan and Shivam not long ago. And we, from the UNESCO, we have, as you know, we have a large program, and all the organizations that you just mentioned are part of of the UNESCO program, including the International Federation of Medical Students Associations. So we had this discussion to include your Rotaract, to have a role unit by itself as we have with others, and so that was how it started. But I'm delighted that you all taken the important area of ethics. You will all, for all of you students who are listening, you should know one of the things is that we brought we launched this bringing ethics into the medical curriculum in 2015 with the president of the then Medical Council of India. And the reason was that every other country, in fact, has ethics in the curriculum. And what we found is all the medical students, as you know, over 2,000 every year go to the developed world or Western world, more than 2,000. And every one of them studied ethics to write the exam to come and work in whether it's US or UK, Australia or Canada or wherever. And so the question was about why is it that you have to do it when you come out, and why not for serving the community and the population wherever you are? And so we had this important area that we launched, and last year with our pushing this came into effect last year. Now that's 21 years after the rest of the world made it mandatory for accreditation. And for all of you to know, from 2022, all those colleges that don't have competency-based education and ethics are not going to be listed in the World Handbook, and that was another reason that this came through. But more important than that, I want to stress here that we've had the best experience with medical students. Right from 2013 when we started, Professor Matthew and Professor Princy were both doing these programs, and it was in Turkey we have the Global Bioethical Inquiry, and one of the first articles by the student was the need to sow the ethical seeds early in our career and our learning, and that sounded very important, and I think that was probably an important aspect to all of us, taking students, giving attention, because we believe that the students, you got your whole career ahead of serving, of caring, and this will make the great difference. Not just that you will be going to practice high quality medicine, but the important aspect is practicing ethical, value based high quality medicine. That's what makes you join the noble profession. If it was only high quality, without the nobility, without the value, and without the ethical part, then it's a business. Okay, and you have all joined the long tradition of a profession that has been for thousands of years has been considered noble. So for that reason, this is very important. I'm delighted that the UNESCO program, the art program, has collaborated when Shubham and Anusha and Keegan met me. I then decided that we will collaborate or cooperate, and so I asked Professor Princy Pallaty, and we then had some discussions. Both of us had some discussions, and I'm very delighted that I have my colleagues, my team, are here with you today and tomorrow and day after. All, including your judges, are all very distinguished colleagues, members of the Indian program of the UNESCO chair. I'm also delighted that Professor Mary Matthew, who heads the Indian program, has joined, and she's going to be there to say a few words, because she was one of the first to start the student wing and taking out way down in 13, 14, taking the students to a school program and several other areas. So there will be a good opportunity to listen to what she says, and probably if we are planning as you wanted to have the Rotaract as part of the Indian program of the UNESCO chair in terms of the student, then I think it will be important that we. I want to, before I forget, I want to congratulate all of you for putting out such a, and Princy, of course, for bringing out such a wonderful program. This is this whole area is of great interest to a lot of people, and we do have in the course, as you know, I've spoken, and to all the medical students, Rotaract, in collaboration with us, with UNESCO chair, we're putting out the international certificate course on bioethics and human rights for medical students. And I know that you're organizing, you will be taking those who are interested to join, meet Subham and Anisha and Kagan, and take advantage of this, because you will have an international faculty from around the world. But one of the modules is on the gene editing from taken on, and so you'll have an introduction not only to this but to other important areas that are coming in very much into which will be very important in your future practice. So with all that, I'm going to wish you all the best, and I will be there and will be again joining you on the for your debate. Is that right, Princy? Is that what you? And I want to thank Dr. Richa, Shailija Mene. I don't know if she's there. They're coming tomorrow. Is that right? Avinash D'Souza, Shilpa Sharma, and Dr. Vishnu, Professor Vishnu Devi, and I think so. What's the from Datamege, isn't it? Is that
Sonali, Dr. Sonali.
Sonali Chowdhury? Is it Sonali? Yes. So is there anyone I left? I want to thank all of them. I spoke to each of them personally and told them that they and requested that they take part in this important program today. So thank you all for joining, and I'll be joining you again for the debate. Okay. So all the best. Congratulations, and Princy, congratulations to you too. I know that you've been calling me and speaking and planning this. So we have also the inauguration of the Asia Pacific Bioethics program. I'm sorry, the World Bioethics Day on Monday, and I think you're all invited to join that, because it will the inauguration. It's being inaugurated by, I don't know if you know, one of the world figures in medical education, Professor Ronald Hardin, the editor of Medical Teacher, and the founder of AMI, the Association of Medical Education for Europe. So he's going to be inaugurating the Asia Pacific celebration, and you are part of the Asia Pacific division, okay? And that will be followed by a stream of another couple of with the student program of the UNESCO with some lectures on the 19th, 21st, and 23rd. Okay, so we have you're all welcome. All of you are welcome. We'll I'll give you. I think by the last day, Princy will put up the link, so those who want, those of you could join. It's 12:30 on Monday, 12:30 in the afternoon, and we'll be getting all the divisions from Asia and Australia joining us on the day. Thank you very much for that.
Thank you so much, sir, for always inspiring us and motivating us to do better in life. We have Dr. Mary Matthew, Head for Indian Program. I will request ma'am to please say some words and inspire the students.
Thank you, Shubham. Can you hear me?
Yes, ma'am, we are.
Yeah. So really, it's such a pleasure and honor to be among all of you. One thing that Dr. Princy and Dr. Russell and I have is passion for our students and to see them do very well. So when the UNESCO chair in bioethics was established here in India, the first thing we did was we started establishing student wings. And we realized that the student wings were the ones who were driving the unit and taking the cause of ethics forward. So right now we have at least 70 student wings all over India, and who are very active. And the World Bioethics Day, which we'll be celebrating on Sunday and on Monday, is when they're going to showcase all their talents and take part in all the programs that is there for the World Bioethics Day. So I encourage all of you to actively join in this unit and take the cause of ethics forward. It's not only for writing the USMLE exams, but also learning about ethics will help you in your future life. So, in your future, rather. So, I congratulate your team for organizing this 3-day webinar, and I wish you all the very best. And I hope you have fun-filled activities, because ethics, usually when you say the word ethics, everyone thinks it's boring, but then we have seen how students can make ethics it's fun to learn and fun to study. And you have the right person, that is Dr. Princy, who's guiding you, and she has been in her capacity as a professor previously in Father Muller. She has taken the lead and taken her students through a lot of programs in bioethics. So congratulations once again, and wish you all the very best. Thank you.
Thank you so much for your inspiring words, ma'am. I believe ethics is a
So,
foundation. Yes, sir. Yes, sir.
I forgot to invite, as Mary said, on Sunday, we have every Sunday we've been having webinars, which is the international panel discussion. This
Yes.
Sunday has been dedicated to the World Bioethics Day, and I want to invite all of you. It's going to be very interesting, because we're starting with the medical students of Prince's College University, who are going to do a dance depicting benefits and harm from an art perspective. And this
Yeah,
is going to be
which is the theme of this year's
World Bioethics Day.
World Bioethics.
So we're going to start with the students performing a dance depicting the benefit and harm theme, but bringing it from an art perspective. And then we have a presentation, and then we have an international panel from around the world who will take on the discussion. So, and the Professor Mary will be moderating, we'll all be there. And this is going to be the 27th of those webinars. I'm sure I think Subham, you've been attending a few of them, isn't it?
Yes, sir. I've been attending all the webinars.
Yeah. So please, we'll put up the link. All of you are welcome to attend the webinar on Sunday, the 18th. It's at 5 o'clock in India, but the times go right around the world. So all of you are welcome to join them.
Thank you so much, sir. Thank you. Thank you so much, sir. Thank you, Mary ma'am, for being with us. It was pleasure having you all today for this session. I would now like to introduce the panelists for today's session. Our esteemed panelists are Dr. Princy Pallaty and Dr. Richa Dayaramani. I would like to mention about Dr. Princy Pallaty. Ma'am is HOD and professor at Department of Pharmacology, School of Medicine, Kochi. Ma'am also had South India Unit UNESCO Chair in Bioethics Haifa, National Chair Curriculum India Program. She has graduated and pursued post-graduation from Goa Medical College. She has worked 5 years at Academy of Medical Sciences, Pariyaram, and 16 years at Father Muller Medical College, Mangalore. Has over 80 indexed peer-reviewed international articles, over 18 books chapters, 2 textbook, reviewer for many journals, on editorial board for Austin Journal of Pharmacology. Uh, she is an avid researcher and received a Rajiv Gandhi University Health Sciences grant prizes total of about 7 lakh rupees, and is involved in medical education and completed basic and advanced course of MCI. Ma'am has interest in bioethics and is the chair for bioethics since 2012 for UNESCO Chair Bioethics Haifa. She has headed the team in preparing curriculum for various disciplines such as pharmacy, medicine, and dental. Our second panelist for today, speaker and panelist for today is Dr. Richa Dayaramani. Ma'am is working as a principal in Khyati College of Pharmacy. She has 20 plus years of professional experience in teaching, research, and industry. She has work experience with Tolani Institute of Pharmacy, IPCA Laboratories, and Risan Pharma Limited. She is a Board of Studies member, Gujarat Technological University syllabus, committee member in Pharmacy Council of India, vice president of APTI Gujarat. She is founder president of Human Ethics Committee Association, committed to empower and propagate ethics in clinical research and clinical trial. She is also the core committee member of UNESCO Bioethics Pharmacy India chapter. She has published 21 research papers in index journals, authored 3 books, and filed three patent. She's presently working specifically for entrepreneurship awareness and development, and has established a nodal center in her college. We are very glad that we have such a amazing panelist for us today. Over to you, ma'am. I will request Dr. Princy, ma'am, to please begin with this session.
Let me thank the organizing committee of these youngsters who have all the energy to put up this show together and quite a short notice. And with the support of Dr. Russell, Dr. Mary, and the whole team, they have really come up with a wonderful program that is, and they have added a new aspect, that is that photo elocution. You would have heard of photo essay and other such. This is photo elocution. So here again, innovation comes. So that's one thing about that. And these students have brought out in this COVID era, they wanted something different. And so they have embarked on the first day we are having CRISPR, and then the next day, tomorrow, we are having artificial intelligence and the ethical considerations. Two latest of the day. That is what the youngsters' heart yearns for. And so here we are to look at the crunch of CRISPR Cas9. And we are looking at, is it a medical technology, and what are the ethical dilemmas that would follow it? Next slide, please. Yeah, this is the, you must be knowing, everyone should know this. If you don't know, it's not fair. This is Watson and Crick, the young scientists in 1951. They discovered their paper was there that brought out the double helix structure of DNA. When was it? Just in the early 1950s. Following up on that, next slide. Following upon that, we started the Human Genome Project, and that lasted for a long duration of time, where the whole gene of a person was imprinted and analyzed. It was there for you, your blueprint, that is there for you to see. Next slide, please. Yeah. You know this guy? Anyone should say? Anybody? Our youngsters, you all have passed through, you all have evolved. Who is he? Charles Darwin. Yes, Charles Darwin. He had brought out the theory of evolution, and he has brought us into the area how we develop, how we evolved, and we evolved through natural selection. Next slide, please. Through natural selection, or you could also say it was random and natural selection that was the key in our evolution. So we as the Homo sapiens have evolved. Although the world has been in existence over 4 billion years, that is there, we have been there only for the past few billions of years, and we have evolved, and that through natural selection, random selection rather, and now we are going into an era of our knowledge. You know, mankind has been curious, and we are finding more and more. And as we know something, we don't sit quiet. Agreed, we are, mankind is called as a rational. Man is a rational animal, man is a social animal. But what triggers them is not just the curiosity. Once the curiosity is filled and you know, then you tend to manipulate those on us and around us for our betterment. And that's where the guided selection has come through. So natural selection thrown to the winds, And now we are coming to a term where we are calling about guided selection. And at this time, I would also like to bring in a new word. You must have heard the word eugenics. Eugenics, yes, for the good genetic makeup. It was a thought that came in the early 1900s, that about in the earlier last turn of the century, how we have to have good genes should be propagated, and so those who are sick, they were forcefully sterilized for this purpose, and all these things were happening. That is eugenics, and one of the negative eugenics was done by Adolf Hitler, as you would remember. And now we come to the term of social Darwinism, that is, making up a type of better society, that's called as social Darwinism. So that's what I would try to tell you, and this is of importance as we go further into our presentation. Next slide, please. Yeah. So here we have the and it is so opportune and it is so coincidental that this year the Nobel Prize was given to Emmanuel and Jennifer for their work in CRISPR. So that's who we have here. This year's Nobel Prize for Chemistry and Medicine. Next slide, please.
I'm sending the.
Yeah. So here we have,
This CRISPR,
here you are seeing into the
student
genome,
program.
the genomic research, how it has gone through. So what do you mean by genomic research? When we talk about genome editing, gene editing, once we have identified the genes, it's all, there are over the same base pairing, ATGC, in whatever way the pairing is the same. And it is so wonderful to note that this pairing can go in so many slices, and the areas that are called as genome, you are aware what is a gene, and the genotype and the phenotype. Okay, so those are the physical characteristics that the genotype would manifest. I will come to that as we get further into the CRISPR technique. So here we have the next slide, please. Yeah. So what is gene editing? So gene editing is done by specific enzymes that cut out the DNA. And this can be done at areas that are targeted, site-specific target areas, and they can be either removed, inserted, or replaced, where there was no, you can add one bit of a gene. Okay, so these are the ways. So they are different methods of doing this. Next slide. Yes, there are many methods of doing it. The older method were the Zens and the Talons. In 1970s, the DNA recombinant technology began. And thereafter, we had gene editing in its rudimentary. I'll show you the history. And you see the Zens and Talons were used before, but they were very costly. They were very time consuming, and it was not very precise. These were the drawbacks. And then came this CRISPR. I'll get you the spelling correctly as we know. I'm delving into the science of gene editing a bit, and then we will go into the turmoil and into the conundrum of ethical aspects. Okay, next slide, please. Yes. So how does the CRISPR work? So despite all the gene editing tools, only the CRISPR Cas9 has made it easy, made it cheaper, made it less time consuming, and it has become very precise. Okay, so what this is doing is, it is a double-stranded nuclease. It breaks the double strand of DNA at the specific site that you need, and it causes non-homologous and homologous insertion, and then there is a repair of the broken DNA. Whenever the DNA strand is broken, your cell goes into a tizzy. It has to repair it in in any way. And when it repairs, it does it, in a haphazard manner. And that is the only question mark in our problem. So it may add or delete a base pair, and that may cause some errors, that could be, I will explain to you with some examples. So here we see how we get knockout and how we get knocking. Okay, next slide, please. That's how we get knockout and knocking. So here we see what the word CRISPR is. Now, when we say CRISPR, it is clustered, regularly interspaced. Clustered, regularly interspaced. Okay. Short chains, they are short chains. So short, palindromic. What is palindrome? Because it starts and begins the same way, like how you have madam or you write Malayalam. It's both ways, the same words in either direction. So that is a palindrome. So palindromic repeats. Okay, that is the word CRISPR. Okay, and the other word that you know is Cas9. What that is a C for CRISPR and AS for associated protein system 9. Okay, so this set goes together. It goes together. CRISPR Cas9 system. So that's why I have explained it. So if I were to tell you, next slide, please. If I were to tell you in a simple manner, it is something like it's a DNA RNA guide. Okay. And this goes on searching this. It has a strand of DNA that is to be checked for. And this strand, your CRISPR Cas9 is looking in the whole strand of DNA and trying to find a match. Is there something similar? If there is something similar, it will go there. And the Cas9 part is the Souza part. And that goes and nicks the DNA strand at that particular location. And there you have the CRISPR Cas9 working, and it either adds on or it removes, and there you have the repair. So that is how CRISPR Cas9 works. So it's a simple enough technique that you can understand. Maybe it is a type of oversimplification, but I'm sure you have understood the process. Now that you have understood the process, you can understand what are the fallouts, what can happen by default. So here we see in the next slide, please. Here we see the various, here I have explained to you what are those short chains and all the aspects of the palindromes and the short chains that are there. Next slide, please. Yeah. Here we see how each of these are going to work and bring out a different aspect. So one of the aspects that we have seen is in children. I will be giving you the picture of Dr. Jankui. You can use it on DNA material, even you can use it on non-DNA free material. You can also use on extra chromosomal DNA like plasmids or phage specific proteins. And the best part of it all, it's not a new discovery. This is something that has been happening regularly and inherent in the bacteria. So the CRISPR first was identified in E. coli, and then it was we did the final things came out when we used the bacteria Streptococcus pyogenes to get the CRISPR, but it was noted way back early in E. coli. And they were finding this palindromic short chains, but they didn't know for what it was. And then they realized that this is the way any bacteria would get immunity to the viruses, because they had this copies of the viral DNA that was made as palindrome repeats. And so whenever another time the bacterial infection came through, this palindromes that they had would guide them and say, oh, there you have the foreign DNA. and that would be attacked. And that is how the bacteria developed adaptive immunity. So CRISPR-Cas9 is an inherent mechanism that a bacteria uses for adaptive immunity. And we have just picked up what is already present naturally, and we have manipulated it to do a host of things which are, yes, under ethical, it's not acceptable, right? Next slide, please. The next slide. Yeah. So here we have a detailed history. So you see, way back in 1987, it was identified in the E. coli, and then you see that it was it was coined there way back in 2000. And you see in 2013, it came to be used, right? The CRISPR-Cas9 came to be used in the one that was taken from Streptococcus pyogenes, and it was used to the best of. In fact, even the COVID vaccine, which is being prepared now, had to use to get the viral strip of for which we need to get antibodies. That viral strip was taken from the viral DNA, RNA. It's a corona is an RNA virus by the CRISPR-Cas9 technique. So is it relevant? Yes, it's relevant today, even in the COVID times. That's what I would like to take. Next slide, please. So with 2013, we see the onslaught of CRISPR-Cas9 going going forth in leaps and bounds, and the applications that it has taken is endless. So here we see how it is adaptation is occurring. It's a little more in. This is how it happens actually in the bacteria. So we see the adaptation, we see the expression, and we see the interference. That's the Cas9 that binds to the DNA. Next. Next, please. Yeah. So we see there are different types of CRISPR systems, and they are the A and the R and the C. Type 1, 2, and 3. Don't break your lovely heads. We are not molecular biologists. I and you need not know about it, but just know there are some types of CRISPR. Next slide, please. Yeah. Now coming to the Cas9 part of it. That is the endonuclease, that is the snipping or the seizure in the whole system. Okay. Next slide, please. So we see there are different types of Cas9 also systems. We have different types of Cas9 systems. Next slide, please. Yes, there are 3 different types of Cas9. As you had 3 different types of CRISPR, these go by the terms as Cas9 D10A, D Cas9, and also we have this first type, right? Next slide, please. So there are types of Cas9 and types of CRISPR. Let's not break our lovely heads on this. Let us go to, this is also how it acts. So it's just a different diagram, but it tells almost similarly, but a little more in depth, that is. So let's go on to the next slide. Yeah. So we see here how these applications of CRISPR-Cas9 can come. They're used for gene silencing. You can use it for transcription repression. You can use it for homologous redefining. We can use it for various other purposes. And one of them is designer babies, which is the most thought of, but it is one way of killing off diseases. It is also used in reverse genetics. Reverse genetics is understanding what this gene, you see the gene, you do not know what it is for. So when you use CRISPR Cas9, alterations, removal, and insertion of this gene will show you phenotypically what are the characteristics that it would induce. So that's how you would know this gene is causing this sort of an effect. That is reverse genetics, and it comes in various other aspects of gene editing, manipulation, genetic engineering. So for all these applications are there, but some of the most sensational ones are in designer babies. I shall come to that a little later. Another aspect that I would like to tell you, next slide please, is how it can inhibit the inherited diseases. So not only in the person himself, it can inherit the inheritable diseases if it is taken off at the germ cell line, at the germ line, maybe in the embryo, or maybe in the sperm, or maybe in the ova. You can, and that will go on, it will be replicated and handed down generations. And we do not know how many generations it will go, but of course, the whole line of descendants would not have, say, Alzheimer's or say any other diseases. In fact, we have this example that I will come to in a little while from now. So here we see the various possibilities that are seen with CRISPR. Next slide, please. One of them is the beta globin. Just a minute, I will tell you about the beta globin in thalassemia. I have another slide coming through. Beta globin in thalassemia could be altered by CRISPR Cas9, and that would eliminate thalassemia. But the child in whom they had done it, the child became better from his thalassemia, but he continued to have developed unknown cancers, unexpectedly developed cancers. Now, this is a gene drive that could be incorporated into the gene edit of a mosquito, and that does not have a malaria carrying capacity or any other vector-borne disease. Now, when you put a gene drive in, that is with the help of with the help of a CRISPR Cas9 genetic editing tool, we can allow it to copy itself. So when this mosquito multiplies, if you just send out 5,000 mosquitoes, you would be able to clear the area of all mosquito carrying, the malaria carrying laden mosquitoes will be decimated. Only these genetically modified ones will be filling the system. But if you had to do it without CRISPR Cas9, it would have taken years, and you would have to synthetically make, if there is 10,000 mosquitoes in an area, you would have to put 100,000. So that was the proportion, and that didn't work. But this kind of a system where you have a gene drive in place with just a few hundreds of mosquitoes soon you would have only this genetically modified mosquitoes. Isn't that something? But no, friends, no, it's not being put to use. No, because of the ethical consideration. I'm building up for that. Okay, next slide, please. So, and this is the explanation that has come for it. So that's why I have already gone through this. So we need a lot. So CRISPR Cas9 makes it easy, less time consuming, and it can repeat itself. It copies and pastes itself, and that's how it can go on repeating. That's why it is called as a gene drive. Next slide, please. So here we see in 1818, Mary Shelley brought out a novel in which there was a young scientist who made Victor Frankenstein made a sapient creature, and that was called against the it was an unorthodox experiment he did. She described and how it got on to the society, so negativity. So that's why when we do anything great major scientific discovery, we should pray that it doesn't become a Frankenstein and hit back at us. So that is the reason why this Frankenstein is a take on these kind of technologies and scientific advancements should not ultimately come and hit back on the society and its moral fabric. Next slide, please. Yeah, this is the case of the beta thalassemia. I said this is the unedited blood picture, and after editing, you see it's all cleared off. The patient is as good as new. And if you were to do this, this was done in a somatic cell. Now, if you were to do it in a germ cell line, then it would be reciprocated down the generations. You would have wiped out all the inherited diseases. Wow, isn't that a wonderful world for us to live in without many of the diseases? And the healthcare costs would be down to the minimum. All the governments and nations would be truly interested. I'm sure they have perked up their ears, right? So this is the kind of thing that we want, and that is why it is being advocated. But then the very scientists who are working in this field are cautious, and they are informing us of the ethical considerations that has to be made before it goes through. Next slide, please. So here we see the safety and the efficacy is of prime importance, and it can be considered in these three aspects, the three aspects that are clearly considered here: safety and efficacy. So if you go through this slide, you are understanding how it would be totally effective. So please note this slide is very, very important. Next, please. Yeah. So how does it differ from the somatic therapy? Somatic therapy is happening, a type of therapy happens on your own cells. Okay. And usually it will be a person who can give consent and is aware of the risks that are inherent and is still giving acceptance for it. So somatic cell editing is all right, but talking about germ cell line editing, that is another question altogether, and we have to be wary of this. Next slide, please. Yeah, yeah. There. The picture was of designer babies, and here comes the question of how we go on to designer babies. So when you choose a baby, or could a parent choose his child, it is a real suspense at the end of 9 months when you get a child. Yeah, this picture do not lose. Yeah. This is Dr. Jian Hui from China, who did a wonderful experiment and continued to do gene editing on human embryo, and he did it on two infants, twins, Lulu and Nana, and altered their uh, altered their gene for preventing HIV. So, and the minute he did it, he thought it was a great breakthrough. He's cooling his heels in the jail. So, friends, these technologies are very good, and let's not be overly over enthusiasm. Let's not go overboard and land up in soup, because we have to be careful when we are doing about human embryos. Next slide, please. Yeah, now coming to designer babies. Okay, so what is a designer baby? Where the genetic modification of choice is chosen in the baby. So you're designing your baby just like you would buy a bike. You youngsters, boys especially, would buy a bike and you customize it, right? So here, even before you get your baby on the line, you are choosing what traits the baby should have, and this can be done because of the gene editing CRISPR Cas9. In fact, it's such an easy technology as opposed to other gene editing techniques that we had of before. This is really easy and less time consuming, and that is why it is being acceptable and being used in every other field. So that is the cause for worry. Okay, so that is the reason why we have to take a step and look back on what we are trying to do. Next slide, please. Yeah. So here we see, and this is what I've told about Janque, about the twins and the infants. Next slide, please. Yeah. So here you choose the points of requirement, and this, there are genes that is telling about your physical characteristics of height, etc. You can prevent diseases like Alzheimer's and stroke. You can choose the color of the skin, color of the eye, have the perfect vision, perfect curly hair, straight hair, color of your hair. Everything can be decided on. But all this comes to ultimately you are making better human beings. You are making better, it has been used in agriculture to use better bananas, better tomatoes, but right now we can make better human beings. That is the question, contentious question that is hanging in balance. Next slide, please. So designer babies is a important aspect that is aligning. I would just like you to think, we are now in 2020. Just take another 15 years. Within 15 years, this is going to become more rampant. Already there are companies. In fact, if you have a lab, there are labs having CRISPR Cas9. They have sent mails to us. I did get a mail. Do you want any work to be done? We can give you the CRISPR. So whatever palindrome you want to be, you want to be copied, just tell them that and they will do it. They have a system and you pay them for it. So there it is. It's here and now. So it's not something that is yet to come. It's here. So please be aware. So just consider, in about 15 years from now, you have a child who has been born through genetic modification, and you have your own child who is not is a natural child, as you would say, and has not undergone genetic modification. So in a society, if you were to think of that time, if many of the people have done the genetic modification, your daughter, as opposed to your neighbor's daughter, who's so beautiful, so tall, so athletic, so intelligent, in fact, more intelligent than you also, if you would care to admit and be honest, and your child who's sniffing away, with all your common infections, but the other neighbor's girl does not have, and she's not as intelligent, but she's so adorable because she's my own, right? So that's how you would consider the genetic modification and these gene editing, and you have these two children. You did not opt for the modification, and your child will be lost because the other children, these super intelligent children, are going to a different school, having a different array, and she will be falling back. And that the child would ask you, why didn't you opt for genetically modified child as others? And maybe it is a question of values. Maybe it is your ethical answer. Maybe it is a question of money, and that's why it was not done. These questions will be there to hit back at you. And even otherwise, the genetically modified child. Now we have GM kids. We are going to have GM kids. Already we are facing it about GM foods. We have GM, genetically modified animals around. Yeah, lab animals also, nude mice, knockout mice. These are all very helpful. But you can have all of these. But the question still arises. We are saying that designer babies and things, we are making better humans. Better? Better for whom? That is the question again. Next slide, please. So here we have, this is another example of how I was just trying to tell you about how it is taken to be. So there you see it. Yeah, you can take a read of this and we'll go on to the next. So uh, next slide, please. Yes. So here we see that we see this adaptive immunity getting on into germline cell editing, how difficult it is, including sperms and ova and zygote. Now, not only that, when we are doing designer babies, it has to be about 15 to 20 or embryos human embryos would be there. And we have finally the best one that has undergone pre-implantation genetic diagnosis. Okay, that has to be, that technique has to be done to ascertain that these formulations have been done. So, so all those human embryos that were there that are viable and well, yeah, we know life begins at birth. And these other human embryos of this, you have taken the one to be furthered and let it go into the pregnancy. The other embryos, what do? You just discard like without a bat of an eyelid? Isn't it another person that we are considering? Isn't it like an abortion? Or what would it be called? Or would you just, it wouldn't affect your conscience that all the other 19 of the embryos are just being discarded? Or is it murder? These are the questions that need to be answered. The next slide, please. Yeah. So I have taken it to the, we can see it, that is we are using this technology for various other purposes. As long as we are using it to correct, we are using it to correct a disorder, a disease, a disability, or we are using it to prevent a disorder or disease or disability, it is fine. But when it comes to enhancement, there the question arises, how much and where does it stop? Where does that question stop? Next slide, please. Yes. So here we see some of the references that I have put through. You can read through. Next slide, please. Because I didn't take all of it into consideration. That's why. So change is really hardest at the beginning, and so the change has already begun. The beginning, it is hard to say, but we are right now in the middle of this gene editing, and it is really messy in the middle. But right at the end, we will see how easy it was once we find out what we ought to do. That is the basis of ethics. We have to see not what is best, but what is right and what we ought to do. So those are the aspects that we see in gene editing that comes out. Next slide, please. Yeah, this is what Dan Kui had done in China for the altering the human embryos for a particular disease. They had tried out with HIV, and we could negate the chance for HIV coming into certain individuals. Next slide, please. Yeah. So we have to think of the ethical angle, and the ethical dimensions of gene editing are plenty, and it has to be thought on the aspects of science, ethics, and governance. And the problem here is governance and the policies are not in place. In fact, all of the countries are quite lax with their policies, but UK has always been quite orthodox and has kept it tight-fisted, but they have given up. In case of human embryos, you can do gene editing if it is correcting a disease. So that kind of a clause has been added. And if you look in the US, you would find that there are no NIH grants for anybody using any gene editing on a human embryo or germ cell line. Any editing of these are taboo, but elsewhere it can be used. Next slide, please. So the tussle is between certain aspects. Next slide, please. It's a tug of war between the scientists and the ethicists, between intervention and enhancement, and between adults who can make a choice for themselves and a prenatal, pre-implantation, prenatal diagnosis and the use for correcting in those. Now, you may have got a child, designer baby of your choice, but again, that child can turn back and tell you, isn't this, I always want curly hair, why did you give me straight hair? Or why did you make my eye color gray, or the cat eyes, green eyes? I would have liked dark eyes. So it's a question of whose like, and who is it, whose life is it anyway? Who is the owner? Who it is that is talking? So it's the human that we are talking about. So we are talking about the person. So that person is not having any say. You are deciding. So these are the factors that will arise. And I have given you examples of intervention and enhancement. Next slide, please. Yeah. So we see we need a moratorium on all of these. The National Academy of Science cell, that is present in Washington DC. It goes on National Academy of Science, Engineering and Medicine. They talk about all the controversial things and they bring down rules and policies for this purpose. And we have also the international policies. There's no governmental, there's no international tie-up and agreement upon this yet. So these are, if you want to work in this field, you should have the governments, the international bodies should take a moratorium. Next slide, please. So that is why many of the countries and the scientists, along with the ethicists, are looking for a public opinion that comes, and that is why they have this window of public opinion that needs to come. And questionnaires are being sent out on these aspects because everyone has a say in this. Because once a designer baby or a gene edited for a particular disease is gone, you may be in for a different kind, or you may be in for something that would change the human species as a whole. And what will happen to the future generations? Future generations is one of the 15th principle that is engraved in the UNESCO Declaration of Bioethics and Human Rights, which was adopted in 2005 on October 19th. That is why we have the World Bioethics Day. And that point also has to be seen. So we have to protect the future generations. Let's not make any mistakes that are incorrigible and irretrievable. So that is the reason why we have to go slow on this breakthrough technology advances that we're having. So we have to understand. And friends, I would like to ask you, since most of our audience today is our students, and that too in the medical field, all of your P and para, all healthcare professionals, including, I will be glad, with Dr. Russell's permission, I would be glad if you would answer a questionnaire on your opinion on all of this. So this is a part of the window of public opinion. Okay, so we could have a research paper in this done by our young friends here, all the opinion of our young friends listening to this. Okay, so that is one of the aspects also that should go into it. Next slide, please. Yeah, so that brings me to the conclusion. But more ethical aspects will be covered by the next panelist who will be talking to us. Richa is going to do a wonderful job. So I stop for now. I talk more on science and a little bit of ethics, and Richa will take us through into ethics a lot more. Thank you.
Thank you so much, ma'am, for briefing us about in detail what is CRISPR-Cas9 gene and its implication and ethical deliberations. We definitely know that there is going to be a lot of disparity when this comes into picture, but then ethics has got a major role to play, and we'll sort it out definitely, ma'am. I would now request Dr. Richa, ma'am, to take over and present her PPT.
Thank you, Dr. Shubham. Thank you, Dr. Princy ma'am, Russell sir, Mary ma'am, for involving me in this wonderful activity of Rotarian Club. Although I am associated with Lions Club for long, and I really like to do things that benefit to the larger of the society. So this is also an amazing kind of awareness program which will uh, brainstorm the young medical students to think in this direction in an ethical manner. So allow me to share my screen. Is my screen visible?
Yes, yes,
Yes.
ma'am. It is visible, ma'am.
Okay. So Princy ma'am is there with me. I hope you're not going anywhere.
No, no, ma'am.
Okay, so most of the things ma'am has already covered, like the in the initial talk regarding the how CRISPR works and all those things. So I'm not going to go into details, but then this is a quick review of the timeline and the advantages and disadvantages of the concept of development of gene editing tools. So before CRISPR-Cas9, we had Talon and zinc fingers, that is abbreviated as ZFN, and which had their own issues. Like zinc finger, it was really difficult to it was off target, and the RNA it was dependent on protein molecules, so it was hard to design those zinc fingers. And then Talon had some technical issues with respect to it was very tedious to work on and things like that. So here comes the CRISPR, which is highly technology oriented, and there is a software that is a free downloadable software that is available, and the kit comes according to your choice. You can order the kit depending on what type of editing you want, and it was showing for some $65 or something. So it's pretty cool to have a thought that here you are and you can edit your own genes for various reasons. But then how in a bioethical perspective, especially when we are talking about the germline gene editing, that is working on the embryos for future offsprings, maybe of we will mostly concentrate on the human aspect, so human offsprings. Then it is a matter of a lot of concern and thought. Careful and mindful thought process is required, whether or not we should take it. So this is a line of difference between somatic gene editing and the germline editing. So somatic gene editing, the research has been permitted. It is permitted by the federal, means US government as well, and Germany also, Europe, many parts of the Europe also. But then germline gene editing, in March 2009, they came up with a document, as ma'am already mentioned, and moratorium has been emphasized on research on germline gene editing, specifically after the whole episode of Dr. He in China. So when it came, So before that, why means I would like to brief upon Dr. He's incident also. As we all know, he was a postdoc student, and this couple approached Dr. Hay, and at that time there was no moratorium. So that CRISPR and gene editing was not banned during that time. In fact, it was taken very positively. But then this doctor, when he was an expert on this gene editing, he had specialized on gene editing, and then when he broke the news that he has edited the embryo with the proper involvement of the couple also, because it was actually proposed by the couple, it was consented by the couple that they want an edited embryo because they feared that HIV would be transmitted to the offspring. So they consented, and rounds of discussions, and and that whole is recorded, and it is with the Science magazine also. They have done it very officially. And then in one of the conferences, he broke up that he has already, the twins are going to, I mean, the babies are going to be born soon. And that's when the whole hue and cry started. And even the person who had designed the CRISPR, or many committee members from China abroad, they also kind of criticized. The tables were turned. So people who were supporting the gene editing, suddenly they got very much concerned about the ethical bioethics, and they everything was blamed on he, as it's reported in many of the documents. And yet China has kept mum, and Dr. He has also not given any statement that he is definitely denying that he has done anything wrong or morally incorrect. But there is published records for this experiment as of now. Till date, we have only one published document which describes the gene editing, or GGE process, germline gene editing process. So the basic risk of this process is the next generation. That this is the matter of concern. In the somatic cells, the edited gene is not passed down to future generations. Whereas in the embryo, since you are doing it on the reproductive cells or the embryo, if the person has children, edited gene is passed on to the future generation. And here comes the whole episode of bioethics concern. So, being very new, lack of evidence, and there are so many diverse opinions from all type of ethnic groups, religions, scientific community. So there are varied opinions. And like say, for example, there are many parts in the world where government is paying to get children. Whereas in India and Asia, we see that we are, sterilizing people. We are motivating them not to have children. Whereas there are many parts of the world where people are paying to get children, to have children. Governments are paying. They are giving some tax benefits and all that. So there is so much of disparity. There is so much of discrepancy, and lot of diversity also, I will say. So therefore, this WHO has released this one. Okay, it is on the authority of the government to decide, or the local or the regional government has to take a call whether they would allow such research or such use of such methods or not. Now, these are the of application of gene editing or gene modification, out of which we would be concentrating the CRISPR technology, with respect to the gene germ editing. Now, the benefits of CRISPR technology in particular is that it is, as I already mentioned, it is just a some dollars, 2-digit dollar value is only available. It is available. Software is available, and it is widely available because it is a software, and it is profitable. It attracts the interest of the communities, of the persons, and then it is innovative. So if somebody is excited with the thought that let me go for a modular baby kind of thing, then they would definitely take a chance. And whereas the disadvantage or the the point of concern is that, or the this is the price gap. Many people, they may fake software would be there, or only the specialized people will know whether they have actually edited the gene or not, or what they have done, because the layman will not understand. And then prohibitively expensive applications it may have, because the software is not costly, but the kit may vary. Now, principles for the govern. Now, here comes our bioethics thing. The principles for the governance of human genome editing. Now, as of now, we believe that any specifically on gene editing should promote well-being. That is the whole concept of research. Otherwise, also, our research should be socially responsible. We believe that. So it has to go in the direction of benefit, providing benefit to everyone and minimizing the harm. It should be transparent. There should be sharing of information and disclosure. Openness should be there. So, and then the stakeholders should be able to understand what's happening to them, why they are the subjects of research, what kind of research is going to be taking place with their consent, and how much autonomy it has to be there. And with the due care, the care of human subjects or the volunteers who are taking part in the study, they should be cared for. They should not be exploited in any whichever manner. But then there should be due care aspect should be there. And any researcher or any scientist or any team should progress in a manner which is evidence based. It should be robust. It should not be a one experiment wonder, but it should be robust enough to be repetitive, reproductive, and reproducible. And then responsible science should be there, because highest standards of research should be adhered to, and it should also be in consensus with the international and professional norms that are prevailing at a particular period of time. There should be respect for persons. That means the dignity of the individuals has to be there. The principle of respect for persons requires recognition for the personal dignity of all the individuals, and the personal choice of individuals should be acknowledged, and there should be respect for individual decisions as well. So all the persons, we consider all people to be equal, to have equal moral value, irrespective of what is their genetic makeup, what is their gender, what is their ethnic roots, anything. But that should be the respect for should also be considered. And there should be fairness in the term that all the cases should be treated alike in the same manner. Equitable distribution of the benefits should be there. And lastly, the seventh is the translational cooperation. So a commitment into collaborative approach to research and governance means the principle of translational cooperation supports a commitment to collaborative approach research and governance. Now, different cultural contexts must be there, are prevailing in our, around the globe. There are different cultures, there are different beliefs, but then we have to respect each and every cultural context. Now, here comes what problems with CRISPR Cas9. As we all understand that it may turn out to be a wonderful tool for the betterment of the society in terms of handling or eliminating many diseases from the human community, Homo sapiens altogether. But then what can be the problems that we may anticipate? So technologically or technically, the CRISPR Cas9, the whole system, there are certain considerations that one has to pay heed to. That is first one, which is also mentioned in the the document or the research document as well, that there may be off target edits. Now we need to ask one question, that okay, the scissors, that means the cas that the guiding RNA that is behaving as a scissor, and when it is going to cut at a particular DNA site, it may do mistake. Human commit mistakes, so a gene may also commit a mistake, because this is like a gun, you shoot. Or maybe I can give you an example of जैसे कई lot of goons are there. So once they एक बार हमने commitment कर दी, अपने आप की भी नहीं सुनते. So once they start, when it is short, then they have to cut something, so they can cut it by mistake also. So how many mistakes are too many? How many of the mistakes can we allow? Can we even allow a single mistake? Yes or no? We can't afford to make any mistake in this. So off target edits is a very big concern, because this guiding RNA can can make a mistake. It could result in a health problem. It can to cancer, might lead to some kind of tumor growth. It suppress some tumor growth. It may cause paralysis, anything. Because the functioning of human body is extremely complex, and I don't think so. This is my purely personal opinion, that no human being is empowered to completely understand, because everything in our body is based on reaction. It is a action reaction thing that is happening in our body. So whenever there is a editing of genes that is taking place, so this is one action, so our body will sooner or later react to it. This is purely my personal opinion. But yes, CRISPR Cas9, the thing is really exciting. So one of the concern is off target edits, how we are going to handle this off target edit. Second point of concern is on target but so how precise this gene editing needs to be? It has to be 100% precise. We can't afford to have a less precise or percentage precision in this. So this is a bigger problem actually than the wrong thing, means than the off-target edit. So what it means is that the processes, as I already mentioned, that many processes in the cell, that is the cell repair processes, are unpredictable. We can 100% we cannot predict what is going to happen, how the cell is going to behave in response. So there may be like say for example, a DNA strand is there and the scissor is cutting. So at the point of cutting, a little bit of DNA, the exact point where it is cut, there may be some changes in those cells and in those genes. Okay, and they may also get affected, means they are not the target genes, they are not the targets, but they may also get affected and they may react. So this is on target. Next, this also, if in case it happens to an embryo, then the whole progeny would be affected. The whole sequence or the family culture, whatever, I don't know what to call this, but the whole offspring process would be affected. Now, wanted but dangerous. So how many edits are safe? That is also one very interesting question. So in 2017, an international efforts, which was headed by this USNA SEM, that is United States National Academies of Science, Engineering and Medicine, they outlined the condition that should be met before editing a human embryo that is destined for implantation. So they have to predetermine the conditions. That means, so one of the criteria in that was DNA sequence created by the edit already be common in the population and carry no risk of disease. So this is playing safe, so you don't have to take any chances with this. So already that DNA sequence has to be fully understood, because who is going to play with the genes? You need to have the understanding of the disease, the genetic progression of the disease, how many genes, where in the genes this disease or the enzyme, the whole process is taking place. So everything should be on the table. Then only you can start with editing of the genes. So unless and until a person is having, or the scientist is having, correct and complete knowledge, which is again a big question mark, because I am a human being, means that the scientist would be a human being, and human beings are subject to so many aspects, which may be ethical, which may not be ethical. So how one is going to determine that? and how many regulations can we actually make, and how we are going to control that. So this remains a big question mark and a point of concern. Am I audible?
Yeah.
Yeah. So
Yes.
yeah. So the whole, the heritable gene editing in people out of reach for the near future would alone remain the requirements. Everybody will want to go for such type of gene editing. That also, maybe it is required, maybe out of excitement, maybe out of innovation, maybe out of curiosity. People may try, mind my words, it is try. It may not be need based, but people may want to try this. Now, the next point of like technological or technical problem with CRISPR Cas9 is patchwork babies. That means mosaics. That the researchers and the scientists can work on this mosaicism and prevent it. So I'll just the whole gene editing process can go rubbish if this mosaicism comes in, because they had initially, they had done it for Huntington's disease in mice, and they were very successful in this. But then there were also certain studies they were published that when an embryo tweaked to correct a gene that causes this particular Huntington's disease, it could contain a mix of corrected and uncorrected cells. So a mix of corrected along with the uncorrected cells, both can be present. Now, for the biopsy, when we are collecting, when they are collecting sample, it can take the uncorrected cell instead of the normal cell. It may collect the mutated cell, which again would pose the problem. So CRISPR-Cas9 is actually inefficient in this with this regards, and it might leave too many cells that are uncorrected to treat the disease, the particular disease that we want to treat. So how much degree of mosaicism can be tolerable, and what are the conditions, and what type of disease we are targeting, it all depends on that. So for example, if we are if we want to treat a liver with gene editing, then if we have a 30% or maybe 40% of the liver edited, then it is okay, because 40% of liver is working and the person can survive. But is something related to eye, or maybe something related to reproductive system or heart, then how much percentage we can tolerate, we can that remains a question and a point of concern. So how much of mosaicism is tolerable, because mosaicism occurs with CRISPR-Cas9 functioning. It is a part of that. Still, we don't have enough technology to completely go away with this patchwork thing. So if having babies already like with such type of patchwork or with such type of this corrections, then the baby itself is okay. One problem maybe the baby is not done with HIV, but it have some other problem, right? So this also becomes a headache for the parents. So who is going to be responsible for that? Who is going is the scientist ready to bear the consequence, or are the parents going to bear the consequence? Who, that is a point of concern. Now, times, testing times, timeline means when we are having a clinical research trial pertaining to genome editing, the design, what are the inclusion exclusion criteria, what is the autonomy of the volunteers that are going to take part in the study, are they really ready, and what should be the design of the trials, what should be the timelines, the washout period, what should be the if there is any, how they need to be addressed, how the serious adverse event need to be addressed, what should be the compensation, and all these things are ambiguous, and we don't have exact answers for such questions. So the more important thing is that this moratorium which is imposed currently. So WHO has concluded in March 2019 that it states it would currently be irresponsible to make heritable edits to the genome in humans. And in Nature magazine, they have called for a global moratorium, and there's members of this US National Academy of Sciences, Medicine, and the Royal Society also. They all unanimously agreed to the fact that we all must have achieved broad societal consensus before making any such decisions of going along with the CRISPR-Cas9. So, researching on CRISPR-Cas9, particularly with the germline gene editing thing. So germline gene editing thing is on moratorium because we can't have with our global, fabric. We can't have. It is very, very difficult to have a global consensus on this issue, and with lot of collaborative research, collateral, bilateral research, it is really it will be on the shoulders of the scientists or the researchers, as happened with JK or the Dr. He. So initially he must be very, very excited, and he must be thinking that he is going to do a breakthrough in science, but ultimately the consequences were very harsh on him. So are other researchers ready to receive such type of comments, such type of reaction? And what about the sponsor? What about the amount of money that goes with the research thing? These are all some points of concern, because deafness, blindness, these are certain areas. Sickle cell anemia, Huntington's disease, these are some diseases where a single gene may be responsible for such type of problems. And deaf or maybe visual impairedness patients, I have seen in my family only, my cousin, he is suffering from this since early childhood. When he was like 2 years old or something, they got to know that he's having a good power, like big power in his eyes, and he was visually and he used to wear the specs, and because of these specs, as in we know in our society, so he used to be made fun of, which he didn't like. But then when he grew up, he was very hesitant to marry, number one. And if they married, but then till date they did not conceive. They purposely kept away to bear any child. So this is one practical example that for a mere not blindness also, this is just a visual impairment. His number was 10 or something like that. He underwent so many surgeries, but they were not successful. But this is a simple example where our society reacts in weird manner, and but still, for such trivial things also, the society is harsh, and it kinds of plays with the thought process and personal person. As ma'am already mentioned in one example, that comparison would be that this edited baby would be very smart, and the natural child would be natural, and how the society will take, and what will be the psychological state of this natural child. These are all also the questions that we need to for the betterment or for the good mental health prevailing in our society. We should also give a thought on such issues that prevail in our society. And this only in India, the world over, we know that racism and things like that, they prevail all over, and they are still on. So people may want that everybody wants to be beautiful, and that is the reason we know that so many creams. As a pharmacist, I know that there is a lot of market for this dermatological treatments, and everybody wants to look beautiful and young, and we want to reverse aging process also, and some work is done on this aging process as well. I know that there are some experiments that are conducted, but actually aging is also very complex, and it is not the response of one cell, but it is a complex methodology that is involved. So maybe expression of aging we can control, but actually we can't control reverse aging. We can control the express aging. Just suppose when we grow old, we know that as we age, our hair will turn white. There will be wrinkles on our face. Maybe there would be the knees would be problematic after a certain age. So some of the organs may be weak, but this is the expression of aging. We can control one or two expressions by gene editing, but actually we know that age happens and we have to be with it. So suppose we control one factor, it may backfire, or it may initiate in response, our body or the cells or the genes in response would initiate another process which we are completely ignorant about. We don't have any idea, because this is not an evidence based treatment, it is a customized treatment. So I can say that I read this very interesting phrase in one of the articles when I was preparing for this presentation today. CRISPR ethics are like uncomfortable bedfellows. So they go hand in hand, but do away with each other, but they have to be with each other. So it is something of this situation. And let us discuss the ethical issues related to the potential failure. There are two kinds of ethical issues that can arise. One due to the potential failure, other due to the success of gene. Suppose this human germline genome editing experiment that a researcher is targeting for a particular couple. What happens if, what are the ethical issues we anticipate for the in response to the failure? And what is this annotation? It is coming. Okay. So ethical issues related to the potential failures, say there are normative frameworks. That means typical, everything is, it is a guided nature of the process, the scientific process or the research. And they address the bioethical principles. We all know that there are the principles of bioethics underlying the clinical research, and the core principles being the autonomy, beneficence, and non-maleficence and justice. And they would be taken into considerations when we are working on human subjects, especially in the field of germline gene editing. Yeah. Okay. So, there are so many, underlying, whenever a person and a scientist is working for the human embryo or a reproductive cell, then what is the policy tool that needs to be adopted with respect to, like, what should be the legislation governing the particular experiment or a particular this one? Please don't send this request for annotation. It confuses me. What should be the regulations, the norms, what should be the professional guidance, because this is all very new. Even Dr. Hay's experiment is so, we don't have, that is why Dr. Hay is under observation. He is not allowed to leave China for a period of 11 years till the babies grow to 12 years, so that they are able to study. Those babies are under study. So because they don't have any, they are clueless what to do. So that is why the whole scientific community pertaining to this particular experiment is in under tight observation. So what should be the legislation, that what should be the regulation, what should be the professional guidance? And because not every researcher is expert, I can say that. There may be few researchers, and this is all again this what we call as a luck factor, or the this factor also works. All the people may be putting on their honest efforts, but some fruit and other do not. And the documents enforcement, what should be the legally binding or self-compliance documents? What should be the if some mistake happens or something is not going as per our principles or the framework or the roadmap, then what would happen? So licensing of that, who will take the responsibility if anything goes wrong? So these are all the ethical issues that should be pondered to in the event of anticipating a potential failure in this GGE thing. And let us now consider or note what are the ethical issues related to the success of human germline genome editing. So, see, ethically, it would be problematic when they function. So, what would be the impact? Because everything is not known, and there are unknown risks that exist with the CRISPR Cas9 research and the use on a large scale. And because there are individuals involved, there are families involved, and then on a broader sense, there are societies involved when we talk about this germline gene editing, because ultimately we will be implanting a a designed individual in the society, and that would gain a lot of attention too. So even we know that if frankly, we live in India, and if we know that my neighbor's son is a test tube baby, uh, I can 100% I can tell you that whenever this person is passing me by, I will have this thought in my mind that he is a test tube baby, and I will observe him with a keen interest. Me being a person of science would also have this thought. Then what should be the reaction of a common a common man in India, full of negativity, full of other things going on the head, and there would be so many of the societal implications of the success of this experiment. Suppose it happens. So these are also the kind of issues that need to be well thought of if we give a green signal to this GGE or when this takes place. Now, main bioethical concern is the eugenics. I'm told that eugenics is the designer baby thing, that we can create an individual of our choice. So this is eugenics. So what eugenics would be to have whatever we desire, what we want our baby to inherit, or like there is a joke I heard in my school days, that Jawaharlal Nehru, and there was a beautiful dancer. So this dancer went to Pandit Jawaharlal Nehru, caused him, okay, let us marry. So Jawaharlal Nehru told that, why do you want to marry me? So she told that, I want that our baby should be as beautiful as myself and as intelligent as you. So Jawaharlal Nehru told that, I am really excited, and I am delighted to receive such a great proposal. But only one concern is there, that if it becomes reverse, that it gets it becomes like it inherits beauty of mine and brain of yours, then what would be the effect? So that is kind of the concern with eugenics, that we are choosing what our should be, and it can be dangerous, it can be Frankenstein or the Prometheus experiment, and it can be disastrous, okay, because the baby would be very, very powerful powerful, and he would know that too also. And many times we can also say that if an embryo, suppose all this work is going to be in the embryo, whether it has the status of an individual or a live human or not, whether the embryo is giving you the consent, okay, you can conduct any experiment on me. No, the parents are taking this decision. So it is the wish of the parents which the embryo, after becoming the individual in this world, will for his life, and maybe with the progeny, with this embryo will, with this new edited human being would be there, passing on to his generation. Okay, how would you classify an embryo, whether an embryo is an individual or not? What type of status, living status the embryo is having? So who is giving the consent? You can't, of course, you can't, ask the embryo whether you want to be edited or not. So it is largely the I can give you one example of this movie, Three Idiots. So the child wanted to be a photographer, but the parents wanted him to be an engineer. And even the child was under pressure because of this choice of career. So imagine if something is done in your body and which the embryo, after becoming the individual, does not approve of. Uh, maybe HIV is not there, but maybe kind of other thing is there. Minor, all the time the abdominal would be paining, for example. Okay, so the embryo would then what, or the maybe the as per the society norms, if the child is bears some not a very beautiful character, then maybe what will be the reaction of the child? Maybe the child does not approve of this particular editing, so he may file a case, or he can sue the parents or not. So this is all, these are all the some points of concern. So in this context also, informed consent becomes a challenge. Informed consent is a challenge in this germline gene editing, which is a very, very important step when we are conducting any clinical trials. What about enhancements? Enhancements, such type of gene editing, I want to be the best, I want to be the best runner, the fastest man on earth. I want to be the, swiftest person. I want to be the tallest. And all these things are possible under the ambit of gene and editing, means germline gene editing. So enhancement of one's capabilities, enhancement of one's beauty, all such type of factors, whether it should be permitted or not, whether it is ethical or not, needs to be thought about. As far as we are using this germline gene for some particular diseases, which the particular scientists would be very, very confident that it is happening because of the mistake of one gene out of the 1 lakh gene sequences that are existing in the human body, it is okay, it may be fine. But when it comes to the open use of such methods, then it needs to be restricted. What about the patent? Because patenting is a time bound process, it is a long lengthy process, it is a costly process, and you can't store the embryo for 20 years. So patenting and safety for that is also a major concern, because it's science, basically it's a research on human beings. Then what happens when there is a situation of incomplete legislations or over legislations? Till now we know that we are having additions or improvisations with our constitution, which before 50 years was thought to be okay, but now after 50 years, with the changing society, with the changing lifestyle, with the change thoughts and beliefs, with the changing culture, we know that something which was okay in the before 50 years may not hold true now. So the legislations, what will be the status of these legislations, what would be the standing of these legislations, and how we can address to this issue, is also something which we need to think about. So this I have discussed. Now because gene therapy involves to the body's set instructions, it raises many unique ethical concerns. So above ethical concerns are the general ethical concerns pertaining to GGE, but certain unique ethical concerns also rise. And there are certain questions that we need to ask. Now, how and bad uses of gene therapy be distinguished? Is there a demarcation that we can put that this is a use of gene therapy and this is the bad use of gene therapy? We can't, because something which is good for you may not be good for me. You want to look fair, is, but it may be not considered that much good when it comes to me. So you can't really have a clear-cut demarcation or a line between the good use of gene therapy and the bad use of gene therapy. Then who decides which traits are normal and which constitute a disability or disorder? we already know that now disabled people, we are we are using the terminology as differently abled. Okay, so that is the thought process, or that is the thing that we are moving to, shifting to rather. So who will decide which traits are normal and which constitute a disability or a disorder? So there would be another question. Suppose I know that, not a question, but it is just coming to my mind while speaking, that I'm having a diabetes and I know that it can transfer to my offspring. So I get this GG done with my embryo, and now my baby would be free from diabetes. We would know it, and uh, he would be very careless with the life, with the eating habits, with the lifestyle, because he would be free, completely free, from acknowledging the fact that because he knows that he will never get a diabetes. Okay, so this all also need to be addressed. Will the high cost of gene therapy make it available only to the wealthy? We all know that only a few percentage of the world is, is not hand to mouth. So, is it okay for for couple to invest or put the sum of money in gene therapy, where the question of making the uh, getting a square meal is also a struggle. So floating such type of uh, medical miracles, is it worth? Then could the widespread use of gene therapy make society less accepting of people who are different? We already discussed this. Should people be allowed to use gene therapy to enhance basic? Also we have discussed. So these are some real-time questions that should be given a thought before advocating the use of CRISPR-Cas9 in humans. Now we know that there are certain prerequisites. These are the technical prerequisites or technical, feasibility of using CRISPR-Cas9, because we know that the success ratio is only somewhere between 11 to 17% only. It is not that you initiate an experiment and you are confident that it would be 100% a success. No, it is not like that. The rate of success is less than 25%. Currently, it may be around, like as I mentioned, 11 to 20%, somewhere around 20%, and with a lot of mosaicism may be present, or there may be some side effects that can be come up as a result of the side effects of the gene or the off target. And then so it is really a difficult decision to take in the current times to make any general predictions regarding the successful strategies and developments of this CRISPR-Cas9. Now, there is second prerequisite is that knowledge of genetic causes and disease and determinants. We should be we should be very exact, and we should be 100% confident that we have complete knowledge about, or the exact cause of the genetic cause of the disease which we want to treat is known to us completely. Because in the absence of this, it can be really disastrous. As we already saw, this Mark Shelley wrote a novel, Prometheus ya to Frankenstein, and that in this book they created this Frankenstein naam ka genetically modified person, and which kind of became disastrous. He turned out to be a monster, and then this monster, this doctor had to die because of this monster. So all these things, need to be thought of, because we know that we are very knowledgeable. There is a different level of knowledge that exists which we don't know. Now the prerequisite 3 is diagnosis of genetic disease. The diagnosis, exact root cause, the exact sequence of the genetic disease. The genetic disease may be complex, it can be simple in the sense there may be, we are exactly knowing that there are 2 or 3 cells that are responsible for expression of such type of genetic disease. And then there may be a complex, like a Alzheimer's disease, it's a completely complex disease. So what is the knowledge quotient about all those things, and how it will be, reflected in the embryo, and how it will, the mutations would be there. Okay, so this also needs to be thought about. Then the fourth prerequisite is the regulatory challenge. So germline editing, whether this is also good or not, because then we will be, not promoting the research unless and until there is a research, then only there would be development. So that is why they were like uncomfortable bedfellows. They go hand in hand, but with lot of arguments. So, there has to be some breakouts for the breakthrough. So these are all the four prerequisites of this research. And again, I leave you all with certain points to ponder. And all of you can go through these questions which still prevail, that triaging, who should have the access to CRISPR technology and the CRISPR products, what should be the extent to which use of CRISPR should be permitted for non-therapeutic purposes, to what extent CRISPR should be permitted in translational and clinical medicine, to what extent should CRISPR experimentation be permitted in basic and preclinical biomedical research. So this part is good. Basic and preclinical biomedical research, it can be to a greater extent the CRISPR experiment permitted, but not to the, that is also completely personal. I'm not a molecular scientist, but yeah, as far as I understand, according to my common man perspective, I have little percentage agreement with later questions. Limiting human genome editing, somatic versus germline editing, and can we limit genome editing to only somatic cells and not permit the research on the germline editing? That also would be wrong, because somewhere some organ out should be there for starting a new treatment. That we as a researcher, as a scientist, we cannot show our back to risk, because whenever we are conducting any scientific experiment, it is a risk that we are going to take. Should international regulations governing CRISPR be crafted and promulgated? So these are the different type of questions or brainstorming that needs to be done before giving a complete nod or a green signal to CRISPR Cas9. So that's all. And this is the one important one-liner that I thought of for this presentation is think before you act. So whenever we want to take a decision, it should be well thought of, specifically when it is relating to the mass population and would be affecting the health and life and the life process of an individual. So thank you, everyone, and I'm open to questions and discussions. Thank you.
Thank you so much, ma'am, for giving us an insight regarding what CRISPR Cas9 technology is and its ethical deliberation. We know that when this comes into picture, there will be fudging of data, there will be disparity among society, and there will be problem regarding informed consent. And any research is considered to be ethical if its benefit outweighs its harm. So definitely this needs to be taken care of well. So without much delay, we'll move on to the panelists. We have gathered some of the questions from the audiences, and we'll be taking some questions from the what we received in the chat box. So over to you, Anusha and Kagan, for the questions to the panelists.
Yes, ma'am. Thank you so much for your presentation. So there's this one question from Aisha Nazreen. That is there any difference in the quality of treatment if we make these corrections as the baby is all grown up and can make their decisions on their own, or is doing it at the germline level more effective?
May I take that question? Yeah. What is the idea of medicine, and what is the best kind of care that you want? You don't even want the expression of any disease to happen even in its mildest symptoms, right? From there, if you want to see from that angle of a disease, correcting it in the germ cell itself is the ideal way of treating, because the patient would never get the symptoms, signs, and there wouldn't be any part of progression. Like now, if you come with hypertension or Alzheimer's, we have to only, halt the progression. We cannot cure the disease, right? So if this can be done right in the early, this is the ideal situation, okay, of therapy. But how we do it and the precision in doing it and the kinds of errors that can creep in, that would be largely, and that is why we need to have ethical deliberations on this. So the question that you that I got, Nazreen, is that should we do gene editing for germline so that we can have the inherited diseases, taken out right from the inception, from the beginning itself? That would be exactly right. But right now, we are not so sure on the technologies. We have to wait a while. But yes, if you're an adult person, I have something and you want to do something, it will be done in my somatic cell line and that correction is allowed. So that is, this is the answer to that question. Somatic
Yeah,
cell
so,
line.
ma'am, with your permission, ma'am, I would like to add one
Yes,
another concept. Ma'am, like the audience, there is also not one embryo that we are researching. When we are researching, it is more or less a kind of test tube baby type of experiment that we are conducting. So there is not a single embryo. So rather than, in my opinion, rather than editing the embryo in question, we can have by selective methodologies, we can have an array of embryos. We can test tube embryos, versions of the embryo, and we can choose, because we already have the Human Genome Project, and we can, diagnose whether the particular responsible gene is present or not. And then we can choose the most appropriate embryo out of all. Because in this manner, the only one, this embryo or one individual would be in question. If we are conducting the genome journal, if we are editing the genes, then this particular thing would pass on to generations, and at present we don't know how it would be amplified or expressed in the future progeny. So this can also be an alternative to addressing this question.
Yeah. One more point of interest is whenever we have a technology that is breakthrough and ready to go, and there is a lot of enthusiasm for it. Let's take the example of test tube baby. It happened in 1987 that the first Louis Brown came, and by 2000 there were 400,000 test tube babies born. Okay. Within a span of just about 10 to 15 years, you had so many babies. So now if you're going to put out this new technology, before even we know what is going to happen or be sure about it, you may be in a big quandary, because once it is out and allowed, we can't put any...
Stop it.
Stopping will be a difficulty. That's it. So that's why we are being cautious right at the beginning.
Thank you so much, ma'am. That's very, very insightful as answers, both Dr. Princy and Dr. Richa. Before we go on to the next question, let me just remind everyone to check their chat box. We have shared the link for certification in the chat box, so please fill that if you want certification. Also, we have shared the link to become a member of the Rotaract Club of Caduceus. So, yeah, if you enjoyed the session, which I'm sure you did, please become a member as well. Moving on to the next question, doctors. Again from Aisha. When we make these small changes at the germ cell level, doesn't it show many unforeseen cascade effects as these organisms / children grow up?
Yes, exactly. That is the point of concern. That is also one of the point of debate. And that is also a point that was considered when this moratorium was put. So, yes, it may have cascading effects.
Right, and we'll go with one last question from the audience. Can CRISPR be helpful in treatment and management of cancer?
Of course, yes. Yes,
It
ma'am,
has
please.
been used. It has been used, but the problem is when you're correcting one thing, I told you, it's not foolproof how the repair will happen. and just dropping a base pair, or you may be you may be uncovering a proto-oncogene that may allow for cancer. So there have been places where you did for beta globin was corrected for thalassemia, but the child had increased tendency for a particular type of cancer. So these are all there. Maybe what we need to know is the refinement in the technology as we see it, and with regulations we can slowly and hesitatingly take little steps forward, slowly and steadily, where we have a controlled situation.
Control.
That's all we can hope for as of now.
Thank you so much. Thank you for those insightful answers. With that, we will end. I see a question from Saurabh in the chat box saying, can you please provide some details of Rotaract Club of Caduceus? So we are an organization that is part of the Rotaract movement. Our motto is putting service over self and actually going on ground and serving the community. Even though we have had this pandemic where we've all been confined to the four walls of our home, we've been doing some wonderful events, and you should definitely become a member. We are an organization, yes. And for membership, Anusha is here, who's the HRD director, who will be able to guide you more about it. So her number is on almost every forum that you would come across. So definitely message her and find out how you can become a member. Great. So with that, I'll hand over to Anusha. Thank you for listening, everyone.
Dear Dr. Princy and Dr. Richa, we'd like to thank you from the bottom of our heart for taking time out for all of us this evening. It's been very insightful, like Keegan said earlier, and we've learned so much through this session. And we really look forward to working with you in the future as well and inviting you again. And I'd like to remind everybody else that yours. That is artificial intelligence reinventing healthcare. And if you did like today's session, do tune in for tomorrow as well. We'd love to host you and take all the questions that you have. Keegan, do you like to add anything?
I
Also,
would like to add something. A special thank to Richa ma'am. I know we gave an invitation in a very short span of time, but ma'am, like I had a call with you, and it was very motivating, to have a call. And you said that, "Nahin, we'll do it. I'll finalize the content and everything. Don't worry, Shubham." So that was so sweet of you to accept invitation within a short span of time and prepare everything so well and present it so well. So a special thanks to you, ma'am, for
So
making
I
out
hope
time
I
for
did
us.
justice with your program, because
Yes,
did,
ma'am,
ma'am, you did
definitely,
more than
Very
justice.
well.
definitely,
Very
definitely.
well done.
And
so
So with that, we will.
and I'm sorry, I
Sorry,
would
ma'am.
like
yes,
to
Go ahead.
ma'am.
specifically mention over here that I'm so glad to see our younger generation who is committed to the society, and being medicos, you are ready to take up uh, new things and working for the benefit of the society, and you are also energetic and so sweet, and I really commend your work. I appreciate all of you, and all the best wishes and blessings to all of you.
Thank you,
Thank you
ma'am.
so much, ma'am.
So much,
Thank you so
ma'am.
much, ma'am.
Ditto.
Thank you. Okay, so with that, we will end our session for today. Thank you, everyone, for joining, especially the panelists. We were so enriched with all of the information that you sent our way. Also, thank you to the participants. We really loved having you and hosting you, and we hope to see you again tomorrow, possibly with 4 or 5 other friends of yours as well, so that the information is not just confined to us and actually spreads out in the community, because that is what we need today.
Yes.
And yeah, to know about the ethical considerations that come with being a doctor. So with that, we will end. I will not take more of your time. Please don't forget to fill the post event form for certification in the chat box, and also become a member. Thank you, everyone.
Thank you. Thank you so much. Thank you so
Thank
much.
you.
So
If possible, can you just share some feedback if it is
Sure,
available
sure,
to you?
yeah,
sure, ma'am. Definitely.
Yes.
yeah.
Thank
And
you. Thank you all.
photograph also.
That
Definitely.
Documentation.
reminds
Yeah.
that reminds
Let's
me.
get a
If everyone can just put their cameras on so that we can take a quick picture.
Yeah, we need everyone to please put on their video so that we can take a smiley photo.
Wait, we have a few screens still white, so if they could put their cameras on, it would be great.
Thank you so much, Princy ma'am, for helping us out with everything.
No problem.
Also, before we forget, I would also like to thank all of the other directors from HRD and International Services who have helped us making this event come possible. And thank you for always being there whenever we needed you to help us. Okay, so with that, we will take a picture.
Shout out to Dhwani, Jalak, Kathleen, Shayan, our vice president, who's here as well.
Yes. Okay, Anusha, do you want to take a picture?
Take it because I'm having issues.
Okay, I'll take it.
We hope to see you again, Richa ma'am.
Oh, thank you. My pleasure.
Shayan, if you want to take some too, please take some because
we are closely associated with Princy ma'am and we go on troubling her with some or other event.
Yeah.
And not to forget a special mention for Dr. Russell. Actually, Russell sir
Yes.
only day before yesterday morning, he mentioned it to me that you have to take part in this program.
Yeah. We asked for some good panelists, but he gave us the best panelist.
Oh, I thought you are going to say we asked for the good panelists and he gave us you. Okay.
Richa and I go a long way back, no, Richa? We have shared
Yes.
the rooms.
Yes, that was a good time. Yeah.
Okay. Good night, all.
Okay. Bye bye.
Thank you.
Good
Thank
night,
you so
everyone.
much.
Bye. Have
Bye.
a nice day.
Bye.